All right, so I'm going in on epithelial tissue again, just because a lot of students seem to really struggle with this section, and histology is not very easy. With this graphic, you have two pictures. There's the actual picture of a slide of under the microscope, and then there's also the simplified version in the graphic. As a student, you're going to have to get used to being able to discern the different formations in the actual microscope slides. So if you look, there's a lot of pink and purple. And so you as a student have to identify what are the distinguishing features, which are the nucleus, the cytoplasm, the cell wall shape and structure, and then noticing the different layers or if it's a single layer. I zoomed in on this next picture so we could draw your attention to the dark purple dots. Those are all nuclei. The pigment of the dye really distinguishes the nucleus for each of the cells, which makes it easy to notice where the actual epithelial cells exist. And so if you see with the simple squamous, they're scattered, and there's also here, the little faint, more lavender versus fuchsia colored. And then over here, these ones are really nice and clearly obvious. You'll always know that columnar cells are columnar cells because the nuclei are all lined up on the basal region, meaning the lower region. And then the upper region, the apical region of the columnar cells are usually going to be a lighter pink or fuchsia color. So another thing I wanted to point out in these pictures is the fact although the graphic to the left that's in beige shows these really clean cells that are in a formated line, that's not how they typically look in reality. So as you see, some of these are like squiggly lines, some of them are round, some of them are kind of just like not very uniform. So what you need to do is to start distinguishing where the surface of the cell is. And why I emphasize the surface is because, as you recall, epithelial tissue has what we call polarity, which it has directional oppositional features. So there's that apical and basal region, which is easily indicated here with the basal region with the dark purple nuclei, and then the fuchsia light pink apical region of this columnar cell. And so every epithelial tissue has an interface or is adjacent or next to what we call a lumen or a free space. So the portion that I'm pointing at here is the free space, otherwise known as the lumen, and this is the empty space or surface that the epithelial tissue is lining up against. So if you think about like on my skin, this free space hovering over my skin is considered the lumen. The free space in my oral cavity is the lumen to the surface of my epithelial tissue of the cheek, of the mucosal lining of my oral cavity. So equivalently, over here in this stomach lining, the white space is the lumen and the surface next to the apical region, and that's how you know that in that free space you might eat a cheeseburger, and this free space is where your food particles are going to be digested and then absorbed through this columnar cell. So that's why directional terms that's why understanding polarity is significant with epithelial tissue. Likewise, if you look at these other pictures here, you'll see that there's free space in these circles because those circles are cross sections of tubules that are found in the kidneys. So in those circles is actually the lumen, and that's where fluid is passing through, and your blood is being filtered, and essentially urine is being produced through these tubules. Likewise, in this picture over here, these are cross sections of these grape hollow-like structures called the alveoli in the lungs. And these spaces are hollow because inside is the free space for air to occupy. So air and oxygen, carbon dioxide are going to diffuse in and out between these simple squamous cells. So you can see that there is a surface for substances, products, air and gas, fluid, whatever it is, to come in contact with this lining of a particular organ. And just since it's here, we have at the bottom the pseudostratified ciliated columnar epithelium, if you look close enough, there's like this little fuzzy border at the top on the apical region. That's the cilia, and that's the finger-like or hair-like projections that sweep away the particles that you inhale that don't get trapped in your nose, but somehow bypass the nasal cavity and now is left for the trachea to try and trap it. Kind of like, I like to equate these cilia like those fly traps that you have at picnics or in your backyard, because they're sticky and they trap particles so that they don't get inhaled further into the lungs. And so in this case, you have the cilia, they get coated with mucin, which is secreted by the cells called the goblet cells that are embedded alongside as neighbors next to the columnar cells down here. So again, there's that free space. If you imagine this whole tissue, think of it vertical and then parallel, so that now you have two sides of it because of its lining the tube of your airway, of your trachea, or your windpipe as we call it lay people. And all of that cilia are sticky with mucus and mucin and trapping all the particulates. And then you like cough or sneeze, and that's why you get like all that stuff that you have to sneeze or blow into a tissue. So again, I just wanted to like emphasize where these tissues might exist, and it's nice that in the visual it indicates on the graphic where you can locate them, because teachers want you to know how to identify them on a slide, the cells, how to identify not just the cell but the tissue type and where they exist in the body, as well as the function. So I just gave you a few examples of how these cells might function and where they are located. So hopefully that is helpful to you. Again, if you have any further questions, I'm glad to answer them. Definitely leave a comment so that I know what exactly I can help you with, because again, most students seem to be asking me help for epithelial tissue. So I'm glad to do that for you guys. See you in the next video.